Happened.
Okay, so let's start our second part of this afternoon session. The first speaker is Fred from currently University of Washington. Actually, the first time I know University of Washington is I watched a movie with Bruce Lee. Yeah, Bruce Lee graduated from University of Washington. When I was very young, I remember. Second time is Fred moved to the University of Washington. Okay, so just a fun joke. Okay, Fred, now go ahead. The floor is yours, and now start your talk. Okay, yeah.
Hello. Since you mentioned it, I couldn't resist saying that Bruce Lee actually is a friend of my family.
Okay.
When he was a kid.
Okay, great.
Of course, we don't have in touch with him for a long time now. Anyway, after he passed away, I thank all the organizers for the opportunity to speak today. It gave me a chance to talk, say publicly, to thank John for the congenial and supportive friendships we had all over the years, all these years, including some collegial collaboration as well. And this is amazing because he actually has so much to do on his hand. All the time he's had to spend on serving the institution, doing outstanding research to advance math and science, and then educating a sizable group of postdocs and graduate students. So I'd like to thank him for that. And Julie and I also wish him on this special occasion, a very happy birth anniversary, 60th birth anniversary, and many more to come. So my talk today is a little bit off from the cancer. I haven't done that for a while. It's on Chlamydia, Chlamydia trachomatis. It's, wait a minute, what happened to my, okay. I didn't move. It's a bacteria that causes different kinds of infections, very serious ones. Among them is an eye infection known as trachoma, some of you might know. And by some estimates, typically about 20 million of people are active with active trachoma, and they cause together roughly 2 million of blindness or severe visual impairment. It's considered the leading cause of irreversible blindness, and the World Health Organization has targeted for eradication by 2020. Well, unfortunately, the data available for 2023 show that this is not happening. And for that reason, this is the reason why there's a continual interest and work on this area to try to learn more about the bacteria. And from what we have learned so far, the bacteria has an unusual 2 days of replication life cycle. And among them, among this unusual form, is that the bacteria exists in two different forms. One of them is called reticulate body, which is short. We use a RB, call it RB for short, and the other one's called elementary bodies, EB for short. The difference between them is that RB divides and proliferates, and it also converts to EB when it wants to, but it perishes, dies outside the host cell. EB, on the other hand, does not divide, does not convert. Once it becomes EB, it stays in EB, but it does survive outside the host cell. So for that reason, it has an interesting evolution in time between the beginning of the RB invasion into the cell, and then gradually convert and eventually perish. And at least the cell lyses and EB survives and go to invade another cell, human cell again. So this is the cycle. Here's a cartoons of that, an EB outside entering a host cell, and it has an inclusion, and it quickly becomes an RB, and then the RB proliferates, divides and proliferates. As soon as somewhere along the line, some of it convert into EB, and this process continues until the whole cell lysis, in which case only the EB survive. So at that point, we have some more information, but the information available until recently has been by conventional electron microscope. It only can take a cross section of the inclusion, and so the data that we get is not very complete, more or less an estimate of the RB and EB at any given time from that cross section, maybe a multiple cross section. But more recently, like our colleague at UCI, Ming Tan and his collaborators, Christine Suderlin, used a novel 3D electronic microscope approach to obtain a 3D view of the inclusion that enclosed all the chlamydia cells, and give us more details about the many other things that we didn't get before, including, of course, the quantification of the temporal change of RB and EB in the inclusion much more accurately. And this is all published in 2018 in an article shown here. And if you're interested, you can get it on my website. So, and here's the image of the three-dimensional view of the inclusion. This is the inclusion, and all the chlamydias sits inside. I'll say more about this later on. For the moment, we want to show a little bit about the time evolution of these bodies, chlamydia bodies. At the beginning, there's only, well, after the invasion into the human cells, the EB quickly become RB, so they're blue, and they're shown in blue here. And as time goes on, after several hours of post invasion, you begin to see some different color, which are the EB. They're smaller in size, and as time goes on, you get this big picture. And I give the time here: 16 hours post invasion, and 28 hours post invasion, and 36 hours post invasion. And the interesting thing is that how does the cell, the bacteria decide when to convert from RB to EB? And I have an idea at the time that this must have something to do with the Darwinian survival of the fittest, and that means that at the end, when at the termination time, that is at the termination time, when the cell lyses, we should have the maximum EB available to go out and survive, and to go invade other human cells. So on the basis of that assumption, I try to formulate a model that describe the situation and determine what the profile for the conversion rate should be. Typically, an R here stands for the population of RBs, and RB grows with natural growth at a rate alpha per unit R, and then you lose them by when it convert to EB. It start with maybe essentially 1 unit, but in any case of RB. So when the EB there's no EB at the beginning, after it comes into the human cells and converted to RB, it accumulated by the conversion from RB over time, and at the end, when at the terminal time t, when the human cell lyses, the total EB is this sum, this integral. So the problem I like to ask myself is, what is the function u of t, the conversion rate, as a function of time that give you the maximum terminal ET, terminal EB, denoted by E sub t. That is the total population of EB at the terminal time, when the cell lyses. When there's all the RB would disappear, only the EB survived. And this is, given the finite energy available, you're not going to have infinite rate of conversion at any given time. So and certainly there's no conversion backwards. That, in other words, EB does not convert it back to RB. So the inequality bound on the conversion rate is between 0 and that's a maximum U. Now the maximum, the U max must be, to make any useful model, the U max must be bigger than alpha. Otherwise, you just convert a maximum rate from the beginning, because you can always have more and more RB to convert at any given time. So so we are only interested in the case where U max is bigger than alpha. So at that point, the maximum principle give us an easy answer. Everything being somewhat linear, you have a bang control, and the optimal conversion rate is nothing, 0. You do nothing for a while, and then a certain point, a certain switching point, TS, time TS, you switch to the maximum rate, conversion rate, and thereafter. And the switching time is determined explicitly in terms of the available parameters, and that's the end of the story in principle. So the question is, how does it compare with the data? And the data looks pretty good. I mean, here's the data available from the first reference I gave by Lee and all, mainly from the Ming-Tan's and Christine Sutherland's labs. So so you can see there's not really nothing happened for 24 hours post invasion, and right away, shortly after that, between 24 and 28, there's a big jump, and so there's the bang controls looks very fitting for this data, and what happened, you can see between 28 and 36, a big change also, and we can see the essentially it looks like a bang control is a good outcome, a good theory for predicting the evolution of the of RB and EB population. However, if you look at it a little more closely, the data tells you a little bit more than what you like to have. but here's a reference that I gave you about the first paper about Ming Tan and Christine Sutherland about the lab work, and there's some theory was included in there as well. The maximum principal effort to determine the conversion rate is reported in this paper with me and Herman and Cecil. So, but if you look at the model I didn't show you before, is the optimal RB population as a function of time. At the beginning, when there's no conversion, the RB grows exponentially, but as soon as you begin the conversion, that's at a raised U max, because U max is bigger than alpha, you're going to get an immediate decline on the RB population, right from the point of switch at the switch time. And so the little cartoon will show you basically here's the bang control. Nothing happened for a while, and then all of a sudden it changed to get converted, and then it get the from that point on, the RB population decline, and then the cell lysis after 48 hours, and you're left with just EB to invade other cells. But as I say before, the actual data shows a little bit different picture in terms of the RB population. It's clear that the EB population has a jump, quite a big jump at the switch point, somewhere between 24 and 28. But the RB population does not decline immediately. It actually went up, and to get a little bit more, you see that the RB population continued to grow from 24 to 28 to 32 hours post invasion. Only after that began to decline. So this does not fit with the prediction of the linear model that we did. And even if you go to a more nonlinear model with, say, logistic growth, right, instead of just linear growth, it does not reproduce this type of outcome, which we did do in the paper that with and Cecil, we did try to do that. We also did some work on the fact that the EB, I mean the EB survive when it lyses, but the lysis time is not the same all the time. So we did a stochastic model, allow the time to be determined by some with some probability distribution, and that does not also lead to any explanation of this growth post conversion growth of the RB population. And so what I'm going to now go back to the experimental result that we have gotten from the first paper. We actually noticed for quite a few other things. For example, the EBs are much smaller than the RB. In fact, you can see the RB, when it divides, it begins to get smaller and smaller. It turns out that only when it gets down to a certain size before it can convert. So the conversion is not, it depends on size as well as the stages of the life cycle. You can see that this, for example, this one is beginning to divide, and is some
excuse me,
smaller size.
Excuse me. Hello?
Division. And when it gets down to this size, then you see that it begin to convert. So
excuse me, Fred. Can I ask a question?
Yeah.
Yeah. I see a lot of brown and green and blue cells. Maybe you can quickly explain what we see in this picture.
Well, the blue one, all the blue ones are the RB pop RB. The red ones, brown, the dark brown and red, is the EB. Now the yellow one, is a somewhat artificial. I think at the time when we published this paper, there was a hypothesis that the RB convert to EB in 2 stages. It first goes to what's called IB and then becomes EB. And that allowed us to try to explain the possible growth of the RB population, total RB population, the sum of all the blue ones, deep blue and light blue and all, different size of blue. When you sum all that up, there's a growth after conversion started. So that was the idea. In fact, it does give an explanation, except the model requires to use an RB sum, the sum of RB at the time of switch, somewhat unrealistic, because not all RB would convert or would divide. That was not separated. So I hope that clarified the situation.
And the entire thing is one cell, or is
one
there
inclusion. One
it's
inclusion.
one
in the cell.
inclusion, I see.
Yeah.
And final question, is this a simulation or a biological image?
Oh, this is a biological image. And this is the one that was enabled to be done by the 3-dimensional electron microscope done by the 10 Sutherland labs. So these are real. I mean, the image from electron microscope, the new technique that they have used to do. Okay, so
can I ask one more? Sorry, just from the picture. So usually there's just one inclusion body per cell?
No, there are more than one inclusion.
I see.
So I'm just showing typically one inclusion this is what happens.
This is one. Okay. And do you have sense of how many there are per cell usually?
Typically about somewhere between around a dozen.
Around a dozen. Okay.
Yeah.
Got it.
It doesn't fill up the whole cell, but the cell has other things. But when the cell lyses, all these things perish. I mean, so that the EB only the EB survived. Okay, so now instead of giving you that 2-stage conversion theory was in one of our papers, and I will not go. So no, that's not okay. In this paper, we were trying to do that, explain that away. But in the end, I didn't think that's really kosher. So I went and do a much more detailed analysis of a multi-stage RB division before conversion. When it gets down to a certain size, before you would convert. So in each stage, you have some of the RB converted into 2 smaller RBs, so called R2, and so on. And only when you get down to a certain stage n-1 is the size of the RB small enough to initiate conversion. So that's what this term is. Okay, so and the EB accumulated as before. Again, we optimize. The optimization problem is to maximize the terminal EB with the constraint of finite conversion rate. And the maximum principle tells you that the conversion rate is bang bang. But now you can add up this total of all the RB of different size, and you get a relationship between the growth of that population. I think this small r is that total r at the switch point, at the switching time. So in that case, you see R prime would be positive if this quantity on the right is positive. and you see that give you a condition between Umax and the growth rate at R alpha n. The difference has to be smaller than the sum in order for that to happen, for the post-conversion growth of the RB population. So while this model tells us that it doesn't always happen, but it would happen if this is true. If the post-conversion RB population would grow for a while when this relationship is valid. So, and apparently the actual bacteria that we have observed must have satisfied this condition. Otherwise, it wouldn't be growing out post conversion. It's a theory that a model that will help explain more realistically why the post conversion growth of the RB population. Whether this is actually the explanation or not, I cannot say for sure, but it does help to ease the worry that there's a difference between the data and the first model that we developed based on a single population conversion. So with that, I think the rest of the details, there are quite a bit of details in those papers that if you're interested, you can take a look. But the idea here is that if you know something about the life cycle, how it works and why it works this way, you might have a chance to intervene and do something about slowing down the replication or proliferation of the Chlamydia population, at least the one that infects the eyes. Thank you. So thank you, and thank you, John, for all years of friendship and collaboration.